Echidnas are one of Australia's most iconic species, yet fundamental questions about their biology and ecology remain unanswered, even 229 years after the species was first named by George Shaw, an English naturalist. Good morning, and welcome to the University of New England. My name is Claire Williams, and today we're going to discuss the potential use of detection dogs in conservation efforts to save the Australian short-beaked echidna. The short-beaked echidna is one of 5 remaining monotremes on Earth. As part of Tachyglossus, the short-beaked echidna shares the same genus with 3 other species of echidna found in New Guinea. Together with the platypus, these 5 species comprise the family Tachyglossidae. As you can see from the diagram on the top right, the echidna and platypus are most closely related to the Australian possums and American opossum. To date, up to 5 subspecies of the short-beaked echidna have been recognized based on varying assessments of geographical, anatomical, and physiological differences. However, for the purposes of this presentation, we will follow the lead of the Zoo and Aquarium Association and recognize just 2 subspecies, those being T.A. acanthion, which is native to the north and west of Australia, and T.A. ecletus, which is native to eastern Australia. But as you can see from the map to the right of the screen, the brown colour shows the range of the short-beaked echidna. So the species has been sighted across the whole of the Australian mainland and Tasmania. So really, we're relying on future advancements in DNA genomics to provide further clarity about how many or if there are any subspecies. As of today, right now, there are just 146 captive specimens worldwide in Zoo and Aquarium Association accredited captive animal facilities. The majority of these, or 82%, are T.A. ecletus. If you look at the numbers on the screen to the right of each subspecies, you'll be able to see the number of each sex of that subspecies currently held in captivity, according to the ZAA Collection Planning online system. So for T.A. ecletus, there are 51 males, 44 females, and 24 of unknown sex. This may seem like quite a large number, but remember, this does not tell us how many of these individuals are mature breeding adults. This number, which is not currently available, is the effective size of the population and is much, much smaller. In fact, it is estimated to be a total of 60 to 70 individuals out of the 146. To date, a number of captive breeding programs have been undertaken with 0 success. No puggle born or hand-raised in captivity has ever survived to become a mature breeding adult. Ever. This is why it is so vitally important to protect our wild populations of short-beaked echidna. The IUCN Red List last assessed the short-beaked echidna population in 2015. At this time, the population was classed as being stable and of least concern for extinction. This does not mean that the species is safe from extinction. Instead, it merely means that the species is at lesser risk than other species are for extinction. The IUCN also noted that there is a continued decline in the number of mature breeding individuals, meaning that effectively that population is decreasing. This decrease is attributed to vehicle collisions as adults, unlike puggles, can protect themselves from predators with their spikes. The main predators of puggles were found to be cats and dogs, both domestic and wild, followed by goannas and pythons. The IUCN also noted that the population is prone to extreme fluctuations, suggesting the entire range needs to be conserved to protect its numbers. If you look at the maps on the slide, the top right first, you can see the data from the New South Wales Bionet Atlas of New South Wales Wildlife, showing around 26,000 sightings of short-beaked echidna in the state. The map below is from the Atlas of Living Australia, showing approximately 40,000 sightings Australia-wide, which includes this 26,000. It's important to note that this data comes from a variety of sources, including citizen scientists, and is not free from omissions, errors, or duplications, and goes back to the start of records before 1900. The short-beaked echidna is commonly found in areas of undisturbed woodland, grassland, and forest, as well as disturbed agricultural lands. The species is typically solitary and quiet in nature and very hard to find. The species mates between June to September, with males forming a train behind a solitary female as they compete for mating rights. The female will then lay a single egg, which she incubates for 3 days in a folded pouch on her stomach before she digs a burrow to house the puggle. The female will return to the burrow around once a week to feed the puggle from milk secreted from mammary glands onto her stomach for about 12 months before the puggle disperses on its own. Typically, the only sign that a burrow exists is the presence of the mother's fecal matter in the close vicinity, usually around 5 to 10 meters from the entrance. We do not currently know what percentage of puggles survive to adulthood. However, we do know that human disturbance can cause a mother to abandon her puggle. Given that no puggle has ever survived in captivity, this is a death sentence. Detection dogs are regularly used to find rare, cryptic, and elusive species, but to date have never been used to locate any species of monotreme. This method of detection is expected to save both time and money, as a human search effort usually reveals 1 burrow per person per 4 hours of search effort. Given the number of valid peer-reviewed studies that have found that dogs can search an area faster, more effectively than humans for a variety of vertebrate species, the use of conservation dogs in the protection of the short-beaked echidna is a potentially successful method to aid conservation efforts. Detection of puggle burrows will allow for protection of surrounding areas from human intervention and removal of invasive species, which will support the puggle and indeed the species' survival. The overarching aim of this project is to determine whether the use of detection dogs is effective in conserving the short-beaked echidna by increasing a puggle's chance of survival in the wild. By locating a puggle burrow, increased surveillance and removal of invasive predators can be undertaken to remove potential threats to the puggle. The primary aim is to determine whether the use of detection dogs aids the search effort for puggle burrows by decreasing search effort compared to human searches. The secondary aim is to determine whether the presence of short-beaked echidna fecal matter predicts the location of a puggle burrow. It is hypothesized that detection dogs will be able to detect the puggle's burrow's presence across multiple habitats up to 100 meters in distance in a period of 15 minutes. To date, there are only 5 known monotreme species in the world, and other than the platypus, only the short-beaked echidna exists in Australia. Being so rare, monotremes offer a great deal of interest to scientists seeking to uncover the evolutionary history of mammals. The short-beaked echidna has not successfully reproduced in any captive environment to date, suggesting that the species is unlikely to do so in the future, and therefore native populations are at even greater risk. Although 14 puggles have been born to date at Currumbin Wildlife Park in partnership with the University of Queensland, none have gone on to survive to maturity. Although current breeding programs are underway at Currumbin, Perth Zoo and Taronga Zoo, with several other facilities looking to acquire the species through such programs, none of these programs has been sustainable or successful to date. In fact, the only echidnas that have ever survived in captivity are weaned adults over 1 year of age. Therefore, the protection of puggles in the native environment is a highly important issue to maintain the population within the environment, conserve its genetics, and ensure its survival. This is important to Australia's biodiversity at the level of genetic, species, and ecosystem. Given the IUCN ranking of the other longbeak echidna species, it is possible that only the short-beaked echidna will survive and avoid extinction. So it is really essential that the genetics of the short short-beaked echidna are saved and banked. Although detection dogs have been utilised for many years detecting a vast array of endemic species, they are yet to be used to detect monotremes such as the short-beaked echidna. Previous programs aiming to protect wild-born puggles from predation have failed based on the amount of time, costs, and human resources involved in such a project. This innovative use of detection dogs aims to increase the number of puggles surviving into adulthood to become mature breeding individuals in their own right. This method offers less direct human interaction with the species, resulting in a decreased stress response in the female and a decreased likelihood that she will abandon the puggle. Although dogs may also elicit this stress response, the increased powers of scent detection mean they can detect a burrow from a greater distance and therefore remain a further distance away from the area than a human could. Conservation detection dogs also abide by strict standards which include the need for the dog to wear boots to minimize scent left in the environment from the paw pads and to be brushed immediately prior to entering the environment to limit the amount of fur left behind. They are also trained to stay away from both animals and their burrows and, as the target owner, the female echidna scat is located at least 5 to 10 meters from the burrow entrance in most cases, they have absolutely no reason to approach either the animal or the burrow. This project will be undertaken by myself, a qualified and experienced zookeeper, detection dog trainer and handler, canine behaviorist, scent work judge, and subject matter expert. I have postgraduate qualifications in conservation and environmental management and canine behavior, as well as being accredited by the Conservation Detection Board of Australia as a detection dog handler for several odours. I am also the author of several published books and articles and have pre-existing relationships with numerous captive animal facilities in order to obtain suitable samples for these trials. This project will follow the methods recommended by Nielsen, Jackson, and Ball in 2016. 2 female, mixed-breed dogs with previous experience in conservation detection roles will be trained to detect short-beaked echidna scat derived from various captive animal facilities. Fecal matter of various states of decomposition will be collected from female echidnas using metal tongs before being placed into sterile glass jars to avoid all potential sources of contamination. The fecal matter will be presented to both dogs as a new scent in the manner recommended by the Conservation Detection Board Certification Board, which is the industry standard for conservation detection dogs. Laboratory training and trials will be conducted at the researcher's place of business before field trials are commenced at habitats where short-beaked echidna commonly reside. Field trials will then be conducted first with scented burrows, but then with natural burrows, or in other words, scat will not be placed in the location the dogs will be finding scat placed by echidnas in their endemic environment. Once each dog is reliably targeting the echidna scat, both will complete the Conservation Detection Dog Certification Board examination. During the final assessment, 30 trials of the same scent will be undertaken over a period of 3 days, with each dog to quantitatively assess whether they can reliably detect the target odor in the environment for a total of 60 trials. The 60 replications will be used to undertake 2 sets of statistics. Firstly, a group analysis using a one-way ANOVA to determine whether a significant difference in the detection rate of puggle burrows between human and canine searches can be found. This will be followed by the association analysis using a regression model to assess whether an association exists between canine scat detection and identification of puggle burrows. The main outcomes from this project will be the answer to 2 main questions. Firstly, we'll know whether dogs can detect puggle burrows more effectively than humans, and secondly, we will know whether there's any relationship between the location of short-beaked echidna scat and puggle burrows. This may be the first workable conservation initiative for the short-beaked echidna. There could be real flow-on effects for conservation of the long-beaked echidna species in New England, which are more at risk than the short-beaked echidna is. This could offer conservationists a real tool to fight for these species' survival. But most of all, we'll have happy echidnas.